at the extant species. Okay. But if you just do that, you're going to miss a whole lot of context. Birds didn't just come out of nowhere. You had millions of years of evolution in their non-avian dinosaur relatives. And then between that earliest bird, you still have 150 million years of evolution there. And so if you only look at modern day birds, you're missing the story of what shaped their brains and what made them the way they are today. Oh, sorry, let me go back 1 minute. So how can we study the brains of extinct birds? Well, one problem with trying to do this is that in the avian fossil record, in the bird fossil record, brains do not preserve. In general, soft tissues don't preserve in fossils. So you can't like go out and go birding and watch these early birds interacting with their environments. You can't directly observe behavior. You can't directly observe brain. And so how are we going to solve this problem? Sorry, my animations are jumping ahead of me. There we go. One option is with something called a brain endocast. So a brain endocast represents the space in which the brain sat during life. So you can think of the bird skull acting as kind of a jello mold, and the brain is the jello. In birds and also in mammals, the shape of that space where the brain sat is actually a really faithful representation of the size and shape of the actual brain. And so we have this pretty good representation of the shape of the brain, and this preserves in the fossil record, because a lot of times you do get bones. Not always, since bird bones are very thin. We don't get a ton of bird fossils where the skull is preserved in 3 dimensions or not broken. But you have a lot more of that than you do of fossilized brains, which are 0. So one way to get a bird brain endocast is with something called CT scanning. It's like a 3D X-ray. As a refresher, X-rays only pick up the dense tissues in your body, so that tends to be like bones and teeth, and they don't pick up air or soft tissues. So you do this 3D X-ray or CT scan of a specimen. It takes a bunch of slices, whereas an X-ray, like if you broke your arm, it's just kind of from front to back, and that's it. A CT scanner takes X-ray images all the way around the specimen, and then you can put those all together to reconstruct the shape of the skull, for example, and the brain that sat inside the skull. But when you see that technology, you might think this is a really new field, like this idea of studying fossil bird brains. It's actually not. It started in the early 1800s because sometimes you would get the shape of the brain preserved in the fossil record, even though you wouldn't have the actual brain preserved. Or researchers started to realize that just looking at the shape of the internal surface of the skull would also look like the brain. But a lot of these early things were kind of just descriptions and also incorrectly assumed that the shape and the size of the bird brain meant that they weren't intelligent. The field really went through a revolution in the 1920s through the 1950s and on, when a scientist named Tilly Edinger started to look at brain endocasts, not just of birds. So here she's looking at a mammal endocast. And I always like to call out Tilly because she has a remarkable story. So her father was a comparative anatomist who also studied brains but didn't look at fossils. She was a Jewish woman who was living in Nazi Germany and was doing research, and was an adult at the time that the Nazis came to power. And her research and the recognition that it had gained internationally was what got her out of Germany and saved her life, basically. So she has letters to friends over at Harvard saying, one way or another, the fossil brains will save me. And they did. She got out. She was able to continue her research. She published on the brain of Archaeopteryx, on things like elephant birds. And she published a correction to a very early paper written by one of the big names in paleontology, where he had said, oh, this is what the brains of these really ancient birds look like. And she went back and looked at the data and realized there were no brains at all. He just kind of made up his own guess of what it would look like. So after Tilly kind of revolutionized the field, a lot more people started to do this. And again, with this new technology, CT scanning, we really got to ramp it up. And so what can studying bird brain endocasts tell us? Well, for one, since we're looking at fossils, we can also look at non-avian dinosaur endocasts and look at the transition from non-avian flightless dinosaurs to birds, because flight is a really complex behavior. And so for a long time, it was hypothesized that it was the acquisition of flight, so birds starting to fly, that drove the expansion of the brain, and especially the thinking portions of the brain, like we call them. So in this figure, if you look at the green portions of the brain, those are the same as the forebrain in us. That's the higher order processing part of the brain. And it expands through time as you get towards birds. But actually, recent research showed that it does expand, but not in a major significant way. The expansion of these regions had actually started before flight started. And so for birds, it might have been that the reason they were able to start flying was because they had this really complex brain that was capable of processing that information. Studying brain endocasts can also shed light on biodiversity and conservation questions, like how accurate is the phrase "dumb as a dodo"? So Dr. Eugenia Gold did work on CT scanning the brain or the skull of a dodo, and she showed that the endocast of the dodo, which is shown in the upper right of this figure of purple brains, didn't differ appreciably in size and shape from their modern day relatives, like pigeons and doves. So they weren't dumb, just unlucky enough to have evolved on an island with no predators that was eventually found by the worst predator of them all, humans. And so when I say like a dodo was as smart as a pigeon, that might not sound like much, but you have to remember that pigeons have managed to coexist with these really dangerous predators, humans, and really like use our landscape. So I think pigeons are actually doing pretty okay on the intelligence scale. So I'm going to dig into some of my data now after that intro, but to kind of reorient us, the endocast, which is shown on the top here, and not the brain, which is shown in cross-section on the bottom, preserved in the avian fossil record. But it's the relative sizes of brain regions and not endocast structures that have been correlated with potential functional capabilities. So that's where that structure and function relationship that I'm interested in is tied. Or to put it another way, brains mediate behavior and endocasts represent brains. So if we can figure out how well endocasts represent brains, then we can actually make inferences about behavior throughout the fossil record and look at how that changed through deep time. So one of the structures I looked at for this, there's going to be some technical terms on this slide. Don't worry, I'll explain them. You don't need to remember the terms themselves. One of the structures I looked at was the optic tectum, which is overlain by the optic lobe on the endocast. Again, sorry for the technical terms, this is just the names. The optic tectum is a structure in the brain that is like a first stop for information from the eyes. So when you receive, we have a similar structure in our brains. When the eyes receive visual information, it goes through the nerves that carry it to the brain, and then the optic tectum birds is the first stop for that information. And the optic tectum is found to be relatively smaller in owls, parrots, ducks, and geese, and birds that definitely have reduction in vision, kakapo and kiwi. So kakapo and kiwi are two nocturnal birds from New Zealand. Kakapo can fly a little bit, kiwi are completely flightless, like we talked about my favorites. And studies on their brains have shown that there's a really significant reduction in this part of the brain that would be processing visual information because they're just not relying on vision as much. Owls have a different approach to that. They got better at getting more visual information with less light. Kiwi and kakapo just said, like, who needs eyes? Who needs to see? and reduced it a lot. So to look at this, I wanted to get a pretty diverse sample of modern day species of birds so that I could then make inferences about extinct birds. So I ended up measuring the surface area of the optic lobe, so the endocast structure, and either collecting published data or measuring the volume of the optic tectum for 34 species of modern birds. And we found that there is a strong and significant relationship between the volume of the optic tectum and the surface area of the optic lobe. So on this plot, you can see each data point is a species of bird. They're all kind of clustering around this line and falling within the confidence intervals, which means that it's showing this strong relationships. So as the optic lobe gets bigger, the endocast structure, the optic tectum, the brain structure, also gets bigger. And so this means we can confidently make inferences about behavior based just on the size of the endocast structure when we don't have that brain structure. Okay. And so then I wanted to turn around and test this out on extinct birds. And so I did a sample where I was looking at the relationship between the optic tectum size, so that brain structure size that's receiving visual information, on the overall brain size, because remember the relative importance of the brain or the behavior is going to be reflected in the relative size of the brain region, not overall, because an ostrich is always going to have a bigger whatever brain region than a hummingbird, for example. We did that and found that a lot of the fossils I looked at, they're these blue dots, they kind of fell where we'd expect them with the sample of the rest of the modern day birds, so the open circles. But there was a weirdo in the modern day ones, the kiwi, again, they keep coming up. And one of the fossils I looked at, Dinornis, or the moa, or at least one of the species of moa. And it's not surprising that the kiwi fell out when you look at the data, kind of fell out outside of the sample set, because there's at least one wild population of kiwi that have actually started to lose their vision. So on this next slide, we can look at the brain endocast of the kiwi and of Dinornis, the moa. And so the moa are a group of, I think we're up to 9 species of extinct flightless birds that also lived in New Zealand. And they went extinct shortly, in a couple hundred years after humans arrived to the island. But you can see there's a person for scale next to a moa skeleton. And so, and on this slide, all of these brains are in the side view and they're facing, well, I don't know if the camera gets flipped. They're facing to the left on the slide. There's a little grouse skull in the bottom to kind of give you an orientation as well. And so this orange section is the part that we're looking at, the optic lobe. In the kiwi, it's so small that you can barely even see it. In Dinornis, it's also pretty small when you compare it to all of these other birds. And so, another human for scale. Here's me next to a moa skeleton at the Carnegie Museum of Natural History. And here's a life depiction of what moa looked like. Their only predator when they evolved on New Zealand was this giant eagle called a Haas eagle. So they're another example, kiwi as well, of evolving on an island without mammalian predators. Kind of disadvantages you once mammals arrive. And so we don't know for sure what this meant for moa behavior. We can't say that they were definitely going blind like that population of kiwi, but that similarity is really striking and is significant. So while we can't say exactly what was going on, we can say that moa had a reduction in their visual system, and specifically in this part of the visual system that's receiving that information from their eyes. So if you guys want to get really technical, this work is published in these 2 papers that I did. You probably don't want to read them, but in case you want to, you can read more about that research. Now I'm going to switch gears to talk about my work at the Science Museum of Minnesota and talk more about museum collections and what we're doing to digitize them. So our biology collection, which I lead, houses about 170,000 specimens of birds, mammals, reptiles, amphibians, fishes, insects, plants, mosses, and mollusks. Think I've got everything there. The collection is mostly from the 1960s and on, but we do have some older things in there, and focuses on the biodiversity of Minnesota and the upper Midwest, but we do have representatives from Antarctica, the Arctic, Mexico, and a few other places around the world. Our specimens are housed in the vault shown here, along with our anthropology and paleontology collections, and they're cared for by me, a collections manager, head of conservation, and a registrar. So it takes a lot to keep these collections going. Like many biology collections at natural history museums, our specimens provide an irreplaceable record of biodiversity in the recent past and how that changes through time. And they're used for research by a wide range of scientists. And my research, like the research I just presented to you, is inextricably tied to museum specimens and the digitization of them. And I know I keep saying that term, I'm going to define it in a minute. On the one hand, CT scanning, so that 3D X-ray, inherently digitizes specimens, like this parrot from an article I co-authored. And on the other hand, the only way I'm able to identify specimens that I want to study at museums around the world is because they have digitized their specimen records in their catalog and made them searchable in online databases like the Smithsonian's database on the right of the slide. So what do I mean by digitization? Digitization can either be capturing any kind of digital record of a specimen or capturing the information associated with that specimen. So you can talk about digitizing specimens and think of digitizing the specimen itself, which is what we're doing by photographing our Minnesota bird and mammal skins for the Bell Museum's Minnesota Biodiversity Atlas. And then digitizing the specimen through photography or like through 3D scans, like CT scans, also digitizes the data that's embodied by the specimen itself, such as anatomy, color, parasite load, and many others. But like I said, digitizing also involves digitizing the records and data documented on specimen tags, which is another part of the work we're doing with the Bell's Minnesota Biodiversity Atlas. This side of digitization forms the backbone for any digitization of the specimens themselves, because you've got to keep the information or the images organized somehow. It helps us direct growth of the collection, so decide what we need to collect next, and digitizes tons of other useful data. And like I said earlier, this is an irreplaceable record of biodiversity. So for example, this specimen was collected in 13th July, 1984. We have the location where it was collected. A lot of times we'll also actually have GPS coordinates on these tags. We can't go back to July 13th, 1984 and know what biodiversity was around in this area at that time. The only way to know is, if someone did really well documented surveys or if we have museum specimens. And so getting that information digitized is really important so that researchers all over can use it to understand biodiversity. And it helps us answer this question that I still don't know the answer to, because we're doing a lot of catch up at the Science Museum. What specimens do we have in our collection? And like I said, this digitized data helps us understand where species occur and how this changes through time and in response to human activities. Those data allow us to make maps like this one, and these maps should be pretty familiar to you if any of you use iNaturalist or eBird or those bird casting things. Those are all mapping biodiversity on the maps that we use to navigate our world. You can make those maps with museum specimens, but again, you can travel back in time and look at that distribution as opposed to, well, you can travel back in time if people uploaded the maps for their checklist, but you can travel back in time for a lot longer than eBird lets you do, for example. Understanding how species have responded to environmental changes in the past is key to mitigating the responses to the changes we're causing now with the climate and in the future. But if you also map out where the world's natural history institutions occur, so that's what these numbers that just appeared on the map are, you'll notice a big disparity between the number of species, which is represented by hot colors here. So you'll see like, huge biodiversity hotspot in South America, for example, and the number of museums. There is also mounting evidence that the people who are most qualified to make attempts to slow the impacts of human-caused changes to the environment are those who live in close relationship with the natural environment, often indigenous peoples, and yet these same people have often been excluded from museums and academic science. So if you're an indigenous person in Brazil who has, through observation studied birds your entire life, there's only going to be 73 museums that have records of that biodiversity on the entire continent where your country is located. That's not a very equitable way to distribute this information. And so this is why we want to digitize it. We want to get it in the hands of anyone who can help with this biodiversity crisis we're going through, who's just interested in it, who feels inspired by knowing where species were and what at different times. So that's one of the main goals of digitizing as well. When it comes to digitizing bird specimens, it's a little tricky because birds can be prepared in 3 major ways. So there's the classic study skins that we saw pictures of earlier with that northern flicker. It's pretty common to make skeletons of birds. So this is what I used in my research for my PhD. And much less common to make fluid preserved specimens of birds. And the grant that I was on as a postdoc in Florida, we were focusing on CT scanning 20,000 fluid preserved specimens of vertebrate animals, not just birds, but birds were a big part of it. And we really had trouble finding these fluid preserved specimens because historically people haven't liked sticking birds in a jar of fluid. They'd much rather make the skin where you can see the plumage. So we tried, CT scanning a study skin just to see could we still get useful data from it? So this slide compares the skeleton of a wet specimen, so a whole fluid preserved bird, or the study skin. This is what they look like when you're doing that cross section, the CT scan. And as we suspected, we found that we cannot get the same kind of data. So with a wet specimen, you can see there's the complete brain case. You can even kind of start to see that shape of the brain. Complete skeleton, you can even see the voice box, the syrinx. In a dry study skin, well, to make those, we take the skeleton out. So there's no skeleton. We actually have to cut into the brain case to remove the brain so that everything can dry out well. And in some of the really old ones, you end up with arsenic deposits, and because they're dense, they also show up on the CT scan along with bones. This isn't to say that you can't digitize study skins. So a lab at Occidental College is doing this amazingly detailed 3D scanning of bird study skins so that you can get things like the plumage. And then they're making them into these interactive online assets that I think are really engaging and really beautiful. So you can actually digitize a study skin too, but you can't get the same information about the internal anatomy as you can from a fluid preserved bird. And another benefit of the fluid preserved birds over skeletons, for example, is that this was one CT scan and you get the whole skeleton together. And sometimes that's really important to understand how the bones fit together and how that changes in different species of birds. Some of my other research that I did was on this new method that was developed in the last, I think maybe really taken off in the last 10 years, where you add a stain to a fluid preserved bird, and that lets you see all these soft tissues that normally you don't get to see on X-ray and CT scans. And that results in some great level of detail. Again, this is published if you want to read more about it with my colleagues at the Smithsonian and Washington University of St. Louis. And as a result, you can get, this is going to be a frog example, but it's still really cool, these 3D models of the soft tissue anatomy of vertebrate animals like frogs or birds. So here's what it would look like without the stain Again, you can only see the skeleton and like an outline of the animal. Once you add the stain, and I hope these animations are working, sometimes they're a little iffy over Zoom. Once you add the stain, you can see the soft tissues, and then you can go through and separate out all these different soft tissue systems. So you can see the muscular system and the circulatory system and the nervous system and the skeleton. You can see how all of these relate together. It becomes a really powerful tool for teaching anatomy, especially when we have to do things virtually, as we've had to do during COVID. So this is another one of the really exciting ways, I think, that digitized museum specimens are being used. So to wrap up a little bit, digitizing museum specimens, to me, inspires creativity and enables cutting-edge research, including my own on the MOA that I presented earlier. It enhances education, especially during a pandemic when we had a lot of remote learning happening, and it introduces learners to natural history collections. I'm not sure how many young people we have on this call, but maybe some people on the call know young people who would be really interested in this online learning activity that the museum, the Science Museum of Minnesota, put together with my former coworkers at the Florida Museum of Natural History called Dinodentist. So we used CT scans of different skulls of modern day species that have different kinds of teeth and of different dinosaurs like Diplodocus to teach kids about, what can we tell from a tooth, just from a tooth, what can we tell about diet? Obviously there's not birds in this because birds don't have teeth, but it is a really fun asset, and I think was a really great example of actually using these digitized museum specimens to do some good. And, at its heart, digitizing museum specimens democratizes access to these irreplaceable records of shared natural history and gets it in the hands of anyone who wants to access it. And so on that note, I really encourage you guys to, if you're interested in this, check out the Bell Museum's Minnesota Biodiversity Atlas. They have an interactive portal. You can search for whatever species you're interested in, and it will pull up the map of where the records occurred, so where it's distributed. And you can also change the date range that you're looking at, for example. So if you wanted to see how, where people were finding common yellow throats between 1970 and 1980, you can filter the data on the Biodiversity Atlas that way to see just those results. So, I'm a little biased. I think it's really fun, but it's a great local resource, and I think you guys should check it out if you're at all interested in this. So with that, I think I'm pretty much ready to wrap up and take any questions. It can be about the research or activities at the museum, or if there are, early career people or young people in the audience who want to know more about how I got to where I am professionally, I'm happy to answer questions like that too.
Thank you,
Okay,
Catherine.
we've got a hand up.
I'll go ahead and take a couple of questions off the chat. Do you consider modern birds dinosaurs? And do you consider birds smart?
Yes to both. So yes, birds are dinosaurs. They are, language is tricky. So when people say they're descended from, I understand where we're coming from. I say it sometimes too, but they are dinosaurs. They are just a certain type of dinosaurs. They're avian dinosaurs. So yes, birds are dinosaurs. So all you birders are really dinosaur nerds. And yes, I would consider birds smart, so we talked about how we understand a lot more about the processing abilities of their brain. We recent research where they basically take a bird brain and a mammal brain and blend them up and count how many neurons are in the blended up parts. When they compare that, birds actually have more neurons for the same size brain than mammals do. I think our problem as humans with trying to talk about intelligence in animals is that we're using our standard of intelligence as if that's the best one, but it's not. That's just the one we have. So animals are intelligent in a lot of ways that we don't understand or are only recently coming to understand, including birds. That's a great
Got it.
question. Oh, two questions, I guess.
Yep. Manley, go ahead and ask yours. You had your hand up. Sorry, I had you on mute. You're going to have to unmute.
Okay, this is a comment. You were just talking about the biodiversity atlas. That is funded from the fund that comes from the state
Yes.
for conservation kinds of things, and some conservatives in the legislature pulled out a number of projects from what had been recommended from the committee and not be funded. And the biodiversity atlas is one of those. So if you're interested in that, contact your state senator and recommend to them the urgency of keeping that. The Bell Museum was basically started to document the natural history of the state. It's celebrating 150 years this year. And this would not be an appropriate time to say, well, we don't need to document that information anymore.
Yeah, that they've been really great partners to work with. So the project had started before I started my job. I moved here in August 2020. And it's been so great to they send students over, so student interns who work in the museum. So it's been really wonderful to watch them learn and appreciate these museum specimens and the importance of the information. So yeah, I would really hate to see that project stall out due to lack of funding. We've really appreciated the support that LCCMR has given the museum, the Science Museum of Minnesota and the Bell Museum in the past. And so we do hope it continues. So thanks for that, Manley.
Just a quick comment to David. No, we do not have to exit the share screen. If you want to put your question in there it is. All right.
I'm going to actually stop sharing my screen so I can see the chat and see everyone else.
You got it. You got it. Perfect.
Yeah. Okay.
I got, how do you collect skins? If somebody has a skin to donate, what's the process?
Okay, so this is a great question. I will tell you how the museum does it, and then I will talk about private citizens. So in order to collect any bird parts, any migratory bird parts, you need an institution needs both a U.S. Fish and Wildlife Migratory Bird Treaty Act permit, and you need a salvage permit for local wildlife from the Minnesota DNR. So we have both of those. Because migratory birds are covered by the Migratory Bird Treaty Act, you cannot technically legally have any part of a bird or their nests. So things that are not migratory birds would be ground dwelling game birds like ruffed grouse or wild turkeys. So they're an exception, but like your waterfowl, even though you can shoot them with a permit, you can't just pick up stuff you find off the side of the road because they're covered by the Migratory Bird Treaty Act. And that information is online. Like if you forget, you're like, what species were on there? The U.S. Fish and Wildlife does publish their list of migratory birds. So we have those permits in place. Technically, private citizens we cannot add you to that permit. However, we've gotten correspondence from Fish and Wildlife and DNR. I haven't, from someone who actually used to be the curator of the birds at the Bell Museum, not the current one, has gotten correspondence that it's not their intention to keep private citizens from helping to contribute to museums. So if you find a dead bird and are able to quickly get in touch with the museum, either the Bell Museum or the Science Museum, we both have these permits in place. Often the museum can take it, and we just treat it as you never had it, like you never owned it because you legally couldn't. You were just stewarding it for the museum. But like I said, quickly is the key. It's got to be very clear that your intent was not to keep it. It's just to go to the museum. If you ever want to donate bird remains to a museum, we need where you found it, like as specific as possible. I actually recommend, if you're tech savvy enough, pull up Google Maps and drop a pin where you are and save that information. Or, you know, if you are at a street address, that's fine too. And we need the date, because without that information, a robin is just a robin. It doesn't help us track change through time and where that change happened. So yes, I cannot officially tell everyone, come find dead birds for us and bring them. But that is the practice in general. If you happen to find them, because it's, you know, no enforcement agency wants what could be useful data for conservation to go to waste. But it's hard. Maybe you aren't familiar with the reason we passed the Migratory Bird Treaty Act is because we were decimating our native bird populations for hats, basically. And other reasons, but like that was a big thing. So there's a good reason these laws are in place. It just makes it a little complicated when you want to do community science, for example, or contribute to museums. That's a great question, and obviously very complicated answer. Okay, Stuart, I see you have a hand up, and then I'll go back to questions in the chat.
Yeah, I do. I had heard or read that woodcocks' brains were upside down. Is that true? And if so, how and why?
So they are not actually upside down, but they are very weirdly shaped. So like if a normal brain is like, you know, my hand is the brain, it's kind of curved like this, woodcock brains kind of go like, whoop, and like tuck all the way up. I can't, there's a lot of why that goes into why a skull is shaped the way it is. And we're evolutionary biologists are still trying to figure that out, because there's a lot of things like, you know, you need to be able to do a lot of stuff with your skull. You need to be able to eat. You need to be able to vocalize. You need to be able to take in sensory information. You need to be able to fit a brain in there. You also need to fit jaw and neck muscles to all these attachments on the skull. For woodcocks, though, one thing that is weird and cool about them is that their orbits, so the holes where their eyeballs are, have actually kind of migrated upwards. And so their field of vision gives them a lot of view of like above and almost like behind them, which is great if you are a bird that other birds like to eat. So what we think is going on is that movement of, well, not, you know, doesn't move during their lifetime, not like flounder, but that migration of the orbits to a more dorsal, like upward position on the woodcock brain kind of required it to reorient everything else. But yeah, so they are super, super weird. They're another one of my favorites. I just really love birds that are like probing because they're very touch sensitive as well. Yeah.
Okay, thanks.
Yeah, you're welcome. Okay, let's see which questions I've missed in the chat.
Smart birds. Are crows and parrots smart? You consider birds smart species?
Yes, crows and parrots is great. There's another paper I was on that I didn't present on tonight. And we're not the only ones who found it. That yes, crows and parrots have relatively larger brains to their body size than all other groups of birds, and they got there through like quickly evolving larger brains. So yes, crows and parrots, you know, they're the examples when you think of smart birds. Crows because of their tool use and able to figure out puzzles and things like that, and parrots for similar reasons and also because of communication and their ability to build a very large vocabulary of human words. So again, we're using our own definition of intelligence, but also if you're looking at the relative size of the brain, crows and parrots are the groups with the biggest size brain in birds. Let's see. Okay, I see a question about, I know museum egg collections cemented shell thinning due to DDT. So yes, they were an important like baseline for this is what shells, the thickness of eggshells before DDT, and this is what they're looking like with DDT. Do you know other examples of conservation data from collections? So I don't know, I don't think I have that specific of an example, which is a great example. An example that I would give of like the way I know that collections data are being used currently by DNR natural resource managers is that, well, one, so our conservation of species it's in there, is based on species. So we make a decision like to protect this species of bird. That's the unit that we're deciding to conserve with the Endangered Species Act. This concept of species is actually defined by museum specimens. So when someone names a new species, in order for it to be a real scientifically recognized species, they have to deposit their example of that species in a museum collection so that anyone else could research it. And so we have a record like, okay, if you want to assign any other animals to this species, it has to be the same as this one that was first named this species. So we can't do conservation of species if we don't even have the records of species. So I think that's like a more esoteric way, but that is a way that museum collections are directly linked to conservation. The more on the ground way is that because we have, we allow people to build ideas of the historic ranges of endangered species, then when we're finally get money and are ready to restore habitats, we can figure out where might be a good place to start. We can figure out what other kinds of organisms need to be in that habitat for them to succeed. So for example, a lot of the prairie butterflies that were around in Minnesota, like the Poweshiek skipperling, the Poweshiek skipperling, as far as we know, is no longer in the state. A lot of the other prairie butterflies are really suffering too. But to restore that habitat, you need to know what kind of plants that butterfly needed to lay its eggs on, what kind of plants that the caterpillars needed to eat, what kind of plants that the butterfly needed to gather nectar from. And so having these records, and I know this is a butterfly example, we're talking birds, but hopefully you can bear with me through it. Having these records of all the different kinds of organisms that occurred at a time when this other organism was there gives conservation managers a place to start for when we want to do restoration of habitat. Or when we want to conserve before it's too late, we can say, well, we really need to focus on these 5 species because this one species that we want to protect depends on all of them. Let's see, can we talk about songbird brains? And then if kiwis are going blind, both from Marge. So songbird brains, I guess I need to know what you want to know about them, Marge. If you want to unmute and give, or you can type it in the chat, a little more information.
Well, I guess, how are they different from the others? So you were showing how the vulture has like the bigger olfactory, I think it was portion of the brain. And so, you know, I'm thinking songbirds, do they have like an area of the brain that's a little more developed to be able to carry the tunes that they do? That kind of thing.
Yes, yeah. So songbird, I thought this might be where you were going, but I wanted to get confirmation. Songbirds do actually have a really developed song circuit, and songbirds and parrots both have what scientists call vocal learning, which is different from just like, you know, being able to repeat things back. Like they are actually learning songs and learning these vocalizations like a language, like we do. Not all species of birds do that. Not many species of mammals do it. So it's actually pretty special about songbirds. Yeah. So yes, they have, it's not as easy to see on their brain as that olfactory bulb on the vulture because it's much deeper, but that circuit that processes their language is much more developed in songbirds than in birds that aren't using this vocal communication as much. Okay, oh, and the kiwi question. So it was only in one population of kiwi that they saw like a couple individuals that were blind. So it could just be that population. It could have just been like 1 or 2, but their eyes are really small relative to the size of their skull, so what you would expect them to be. If they will go extinct soonish, I hope not, but they've needed a lot of help to hang on. So New Zealand has really stringent laws to protect them. If I remember correctly, if your pet, like dog or cat, gets out and kills a kiwi, it's getting euthanized, no questions. They have really stringent laws about, you know, preventing poaching or export. And actually, I can't remember the time frame for it, but for a while, New Zealand was not sending any kiwi to at least US zoos and potentially other zoos because we weren't being very good partners about like sending things back. Kiwi are also culturally significant to some of the Maori people. And so now they finally, actually, the person at the Smithsonian Zoo was the one who kind of started the Kiwi exchange again with zoos. She's the one who keeps the stud book for all the Kiwi populations in US zoos. And she built that relationship. And so now there's rules where if it was a Kiwi that was first generation into a zoo, so it was born in the wild, its remains have to be sent back to Maori people. I think it's also second generation they go back, and only third generation the remains can start to go into museum collections. Sorry, this is getting really like Kiwi. We're getting into long-winded answers. The reason they were doing okay before, you know, mammalian predators came along was because yes, they can't see, but they have really sensitive bill tips. So they're another one of those groups that have that part of their brain that processes touch information from the bill is really enlarged. And they also have a really good sense of smell for birds. So they had two other senses that they decided to really invest in, and to the point that they kind of de-invested in their visual senses. Thanks for giving me a chance to talk about kiwi. Let's see, how about song development in songbirds? David, are you talking about how like some songbirds, they might learn a different species song if they're raised with that? Sure. So I don't have great answers to that question because all the birds that I have studied for my research are already dead. I've never really gotten to like the development side. So sorry, okay, I can't help out with that one. And then the very deep philosophical question, how do cowbirds know they are cowbirds? I don't know that avian neurologists have tried to like look for correlates of a sense of self and self-awareness in birds. I don't, you know, all these ideas of species are things that we scientists are putting on to birds too. They know some of the boundaries because they don't tend to breed too far out of their close relatives or even, you know, out of their own species. But I don't think a cowbird really really knows that it is our definition of a cowbird.
Anyone else? Your one chance to talk about bird and dinosaur brains.
Yeah. Okay. I like this hypothesis with Barbara. Like maybe they don't know until they have to put it to the test. I mean, they know generally who to go with because they respond to the signals within their same species. Wow, this is getting really deep. we could go a lot of ways with this. Yeah, but I'm happy to answer any more questions. You guys have been a great audience. Thank you for joining me.
This was great. Thank you.
Yeah, thank you very much, Catherine.
Have a good week, and everybody enjoy the warblers. They're thick and heavy, and
Yeah, they're really coming.
this is we're in peak week.
Yes.
All right. Well, if you all want to unmute and give a round of applause to Dr. Early, thank you very much.
True. Thank you.
Thank you, Catherine. Yeah.
Can I unmute all in one swipe? I don't know. Anyways, thank you, Catherine.
Yeah. Thank you. Well, I'll hang out and chat with the organizers real quick.
There you go.
Everyone else, thank you so much for joining me. And please do come to the Science Museum and see, you know, we don't have a lot of birds on display, but we've got the biodiversity stuff out there and plenty of dinosaurs too.
Okay.
Catherine, I have a question unrelated to birds. I was just wondering, how did well, sort of related, I guess. How did the Science Museum of Minnesota get so many dinosaur skeletons when Minnesota actually doesn't have that many dinosaurs?
Have you heard the name Bruce Erickson?
I suppose he was a paleontologist or is
Yes,
a paleontologist
he
at your
was.
museum.
Let me find, he passed away recently.
Okay.
Let me find his obituary. for you because it goes into detail. Wait, this is not the one I wanted. There's like a full story. He joined the Science Museum early on, like it might have been in the 50s, and his promise was to bring dinosaurs to the museum. So he would go out west where there are more dinosaurs, and he made good on his promise. I think the big one he brought in was the Triceratops. It was the first Triceratops in the state.
That's right.
Yeah. So here, I found the article, so I'll put it in the chat if anyone wants to hear the amazing story of how we got so many dinosaurs. He was just a prolific collector, and he worked at the Science Museum for a long time. I think he was at the Science Museum for like 50 something years.
Okay, that's a long time.
Yes, yeah. So.
Hired in 1959 to go find some dinosaurs. Wow.
They knew dinosaurs bring people in, and so they wanted someone who would do it, and he did. So we now, Bruce.
Oh, this is just
retired.
recently, yeah.
Yeah, he passed away recently. He retired a little bit longer ago than that because we have a current curator of paleontology who continues the legacy of fieldwork and of studying a lot of the reptiles that Bruce found in an area called the Wanagon Creek Formation. So our current curator of paleontology is really into like turtles and crocodiles. Crocodilians are his favorite, and snakes. So it was a pretty good fit for him to step into this collection that Bruce had built, along with the dinosaurs, of course.
Very nice, thank you.
That's cool.
And I guess if we're talking about people who shaped museums, the person in whose honor my position is endowed, like the name Barbara Brown.
Yeah.
Yeah, she was a huge birder in Chicago, and a big supporter of the Field Museum of Natural History. And she would actually go out on field expeditions with the mammal department. I'm not sure why she never went out birds. Maybe she liked looking but not killing them. But she would go out with the mammal department and go on expeditions and prepare specimens. So it's kind of nice for her legacy to be carried on at a different museum now too.
You mentioned in your talk that you appreciate having the bird specimens preserved in fluid rather than skins. And it was really, I never really understood why that was important, but yeah, that's really clear to see now. Thank you for showing that.
Oh, good. I was worried that would get a little too boring. So
no, but are you preserving the birds now in fluid or primarily skeletons?
So we still, for now, we have plenty of room to put fluid specimens. But for now, we're still mostly doing skins or skeletons because that's what we have the capacity for. So there wasn't a curator of biology for about 20 years before I started. It was just something previous administration had decided, and then it kind of just fell to the wayside. And now current president was the one who led the effort to get this position created. So I'm always playing a lot of catch up along with my collections manager, who actually contributed a lot of bird specimens to the Bell Museum as well, because he worked there, and then he's worked at the Science Museum since 1986. But sometimes we'll do fluid ones. We just haven't, like, we need new jars to do it, right? And we need to have time to sit down and refresh on the policy. But he can make a study scan. Like, we found a song sparrow outside the museum last week that it must have hit the glass and died. And he had that from a carcass with all the wet parts. It turned into a study skin in 20 minutes.
Oh, that's pretty fast.
Yeah. He's an expert at doing study skins. And if you come to the museum, you can see our other hard workers in the biology department. We keep a colony of flesh-eating beetles, dermestid beetles. And so they're the ones who do the skeletonizing. So if we want something turned into a skeleton, we take off the skin, take out the guts, and take off, depending on how big it is, a lot of the muscle. but leave some soft tissue. And then we put it in the dermestid colony, and you can actually see the dermestid colony from the exhibit floor. It's kind of tucked away, but if you're down on the third, yeah, the bottom floor of the exhibits where the dinosaurs and fossils are, there's some large mounts of like a polar bear and a brown bear. Across from them, there's a little window into the dermestids. And they've been really happy recently because we just dissected a mountain lion that was hit by a car in Bloomington.
Oh, wow.
Yeah.
So there's a lot of meat on that for them to eat.
Mhm.
Yeah,
Huh.
cool.
When you preserve a bird in fluid, does the fluid affect the plumage, the color or otherwise?
Yeah, so it does. Like if you think about structural color, that kind of depends on how light's refracted, and that's different through fluid. Actual colors like orange colors, carotenoids, they tend to leach out in fluid.
Okay.
So it's not a perfect preservation medium for every usage. Just like a study skin is great for studying plumage, but not great for studying internal anatomy. You know, the fluid preserved things aren't going to be great for looking at plumage.
Okay.
So there's always a trade-off when you decide how to preserve something.
Is the fluid like a formaldehyde or what?
Yeah, so it's called a fixation and then preservation. The fixation happens in formalin, and then we kind of rinse that out because it's done its work on the soft tissues, and then we preserve it in 70% ethanol. Yeah, so we have all of our fluid specimens are in a separate room where we have like graded floor in case, like floor that is grates in case we drop anything. We don't have a big wet mess. Better fire protection because we have great fire suppression for the whole collection, but we have extra for that room because ethanol is flammable. So
my
the fun fact about the Smithsonian, they don't have their fluid preserved specimens in their main museum because no one wanted a giant collection of flammable fluids on the National Mall. So they're actually off-site at a location in Maryland.
Gee, I
Barbara,
wonder why.
okay. Wait, what did you say, Barbara?
Well, I was just going to say that my husband was the education director at the Science Museum for 3 years in the 60s. And he
Cool.
loved having lunch with Bruce.
Oh yeah,
And
Bruce
he
was.
said
I
it was
didn't
like
get to
it
meet
was like
him.
a lecture it was like a fascinating workshop learning experience every lunch hour.
Yes,
He
yeah.
loved Bruce.
He built a really impressive core of volunteers too, who are still coming in. I was just disappointed that I didn't get to meet him, but I moved during the pandemic, so that wasn't a good time for meeting strangers.
And I don't know if you know, but that one big dinosaur was found by those students. There was a student group that found it. That was my husband's school, Anoka High School student.
Oh, that's so cool. I didn't know that.
Very proud of it.
Yes, obviously.
I can't remember which one it was. It wasn't the Triceratops, but
Okay.
it was later on. It was in the 60s, I think, in Wyoming.
Oh,
Anyway, he liked the Science Museum and he loved being out at Warner all summer.
Oh yeah. Yeah, we have a lot of actually one of our entomology volunteer who has been volunteering with the Science Museum for 57 years, which is incredible. He's the reason I know what I know about butterflies in Minnesota. I think he used to carpool with Bruce. Like it was just Bruce really made a community at the museum.
They did. Anyway, thank you for your information. I enjoyed hearing about it. And
yes,
if I find any skin specimens, I'll rush them right over.
But make sure you write down where and when you found it.
You don't really want any more, really? I mean, you've got buildings full of them. Or
no, we do.
Specific?
So this is a common misconception. I mean, we don't want everything that dies ever.
Yeah, every sparrow and
right. But I think a lot of people, see that picture in the last slide of my talk where it's just drawers and drawers and drawers of dead birds and think, well, we don't need to collect anymore. But an article came out recently, and we've known this for a while now, that collecting currently has precipitously declined And that's actually doing a huge disservice to future generations of scientists, because we're going to have data from, the 70s, 80s, 90s,
And
and
then,
maybe earlier, but then not have data about now when we're in a biodiversity crisis. So there's been a big call for more people to do collecting, but museum collections are generally underfunded. And of course, there's permits. We do international travel. It's complicated too. And so like, I don't have time right now to set up a whole new collecting program. That's why we're just taking things in by salvage while I'm still kind of getting things on online and getting things organized. But yeah, just because we have a lot of things doesn't mean we shouldn't be collecting in the future, because we don't want to miss the snapshots of what's happening right now.
okay, well, then I won't discourage people from doing that. Does the Science Museum, do you have bird decals or something on your windows? That help with the
I
pollution?
don't know.
You
I
should
don't know if we
check that.
do.
You could check that.
Yeah, that's a good idea.
One of the reasons for collecting, taking species that people find is that they may not turn out to be what the person thought. I have a friend who picked up, this was in Illinois, 4 yellow warblers and took them to a museum. It turned out one was a hybrid with an Austin warbler. And so the museum is glad to get it, kind of thing.
Well, and I mean, I don't know how obvious it was, but I would guess that might not be something you wouldn't be able to notice if you were watching the bird flying around. You kind of have to observe it up close as a specimen.
Sure.
So yeah, that's cool.
Thank you, Catherine.
You're
Yes.
welcome.
Thank
Thanks
you.
for staying on for more
Thank you.
questions.
Thank you very much.
Meeting you in person someday soon.
Yeah.
Yes, someday.
Yeah, we'll have this up on our St. Paul Audubon YouTube channel tomorrow. I apologize. I forgot about it at the beginning and missed the first 10 minutes. So
Oh,
I
that's
missed your
okay.
introduction, but we got your spiel, so that should be good.
Okay.
I'll send you that, get that link out there at some point.
Thank you,
All right.
Catherine.
Thank you, everybody.
Bye-bye.
Have a good night.
Good night.